Hello, we are team number 5, and today we will talk about the cell transport. First of all, let's give a little context, remembering that the cell is the smallest unit of life. It's the basic structural and functional unit of all living things. This one contains all the genetic material. Now, according to this, the cell has some substance in it, and this one has to move the substance across the body. This process we call it cell transport. In this presentation, we will see the types and examples.
Is the fundamental movement of ions and other molecular substances within the cells along the concentration gradient, without any external energy. It is also known as passive diffusion. There are four types of passive transport: simple diffusion, facilitated diffusion, filtration, and osmosis. Here are some examples. Ethanol enters our bodies and hits the bloodstream. This happens because the ethanol molecules undergo simple diffusion and pass through the cell membrane without any external energy. Reabsorption of nutrients by the intestines by separating them from the solid waste and transporting the nutrients through the intestinal membrane into the bloodstream. And the last one, when a resin is soaked in water, the water moves inside the resin by the process of osmosis and it swells. What is active transport? Is defined as a process that involves the movement of molecules from a region of lower concentration to a region of a higher concentration against a gradient or an obstacle with the use of external energy. During the process of active transport, a protein pump makes use of stored energy in the form of ATP to move molecules. Active transport in plants. Like humans and animals, plants also require a transport system which are mainly involved in transport of materials such as water, minerals, and necessary nutrients to all parts of the plants for its survival. Here are some examples of active transport: movement of Ca2 plus ions out of cardiac muscle cells, transportation of amino acids across the intestinal lining into human gut, and the last one, secretion of proteins like enzymes, peptides, hormones, and antibodies from different cells.
The facilitated diffusion is a passive movement of molecules from the region of higher concentration to the region of lower concentration. It's a selective process because the membrane allows only selected molecules and ions to pass through it. Some factors that can be affecting the facilitated diffusion is the temperature, concentration, diffusion distance, and the size of the molecules. The temperature can be affected the facilitated diffusion because when the temperature increases the movement of molecules too. The concentration is the movement of molecules takes place from the region of higher concentration to lower concentration. And the size of molecules is because the smaller molecules are lighter and hence diffuse faster than the lighter molecules. Some example of this could be the glucose transporter and ion channels. Diffusion is the process of movement of molecules under a concentration gradient. It's an important process occurring in all living beings. Diffusion helps in the movement of substances in and out of cells. The molecules move from a region of higher concentration to a region of lower concentration. Some examples is, for example, take water in a beaker, and diffusion have types, that is simple diffusion and facilitated diffusion. The simple diffusion is a process in which the substance moves through a semi-permeable membrane or in a solution without any help from transportation proteins. The factors that can be affecting the diffusion is the temperature and the size of the particles. And some example of diffusion could be the tea, perfume, food coloring, breathing, cigarette smoke, etc.
Osmosis. What is exactly osmosis? Well, this is the process of water movement through a semi-permeable membrane from an area of lower solute concentration to an area of higher solute concentration until the solute concentration are equalized on both sides of the membrane. Well, in this image, we can see osmosis because it is the movement of water molecules through a semi-permeable membrane. It moves from an area of low solute concentration to an area with high solute concentration, trying to balance both sides. A characteristic of the semi-permeable membrane is that allows certain substances, like water, to pass through, but it blocks others that are larger, like solids. An example of osmosis is the cell membrane, which control the exchange of water and nutrients. In the next slide, we have that osmosis divides into three types of solution. Hypotonic, that the solute concentration is lower outside the cell than inside. Water enters the cell. Hypertonic, that the solute concentration is higher outside the cell than inside. Water leaves the cell. And isotonic, the solution concentration is equal on both sides of the membrane. No yet, movement of water occurs. In the image, we have hypotonic solution that net water gain cell swells, hypertonic solution that net water loss cell shrinks, and isotonic solution not less loss or gain. So an example of this is when a people add salt to a snail or sloth, and this causes the animal lose the water that has in the body due to the salt. The applications could be in the medicine because the balance of fluids and electrolysis in the body depends on osmosis to maintain proper cellular functions.
Active transport is when a certain movement of molecules or ions across the membrane against a concentration that requires cell energy. This movement goes to an area of low concentration to a place with high concentration. Then the phagocytosis is when a cell extends part of its membrane and forms pseudopods and envelopes the particles and encloses them in a membranous pocket called a digestive vacuole. Then these cellular actives are pinocytosis, that is the first image that you can see. Then the phagocytosis, that is the second one, and then the receptor mediate, that is the third one. Then all of these has the extracellular fluid, that is the first part. Then the plasma membrane, that is the line that is below the extracellular fluid, and then at the bottom is the cytoplasms. Then the pinocytosis is the cellular globs in an extracellular fluid, and its dissolved contains not specifically. Then the phagocytosis is the cell envelopes large particles by extending its membrane outward. This creates a large vesicle called a phagosome. Then the final, that is the receptor mediated, a very specific process, that is the cell use receptors on its surface to bind to desired molecules. This triggers the membrane to invaginate and form a vesicle containing a concentrated dose of the target molecule. This is much more efficient than simply grouping in everything.
At least, but not last, we have all the processes that needs a lot of energy to release substances. These are the endocytosis and exocytosis. The first thing that we need to know to understand the meaning is the acronyms of the words endo and exo. Endo, that means inside, and exo, that means outside, relating that with the processes that proteins do. So, endocytosis is when the cells take in substance from outside by involving them in a vesicle derived from the cell membrane. And exocytosis is the process in which proteins are stored in vesicles from transport to subsequent diffusion to the outside from the membrane, enclosing in vesicles to diffuse their contents out of the cortisol. So, in the next slide, we're going to see a very broad graphical explanation of what we just learned from this topic. We hope you have understood this topic perfectly. If you have doubts, do not hesitate to ask. Thank you for your attention. Bye.